Okay, hello all. This week we're going to be looking at different plant tissues. One thing to note before I dive into what we're going to be doing is that your plant diversity and tree thinking write-up is due today, so make sure you bring that in printed to class. All right, so this week we're going to be looking at plant tissues. Before we dive into the specific goals of this lab, I'll give you a brief introduction to tissue systems and the different cell types that you'll see in lab, since I know that we probably won't get there in lecture. However, you will, and Dave will help explain this when he does. Okay, so just a reminder, we've been focused in on the green plants, so that's green algae and land plants in the group Plantae. If we zoom in a little bit more, this phylogeny might look a little bit more familiar to you. And so these are all of the different plant groups that we looked at last week. And so we have our green algae, our non-vascular plants, so our liverworts, hornworts, and mosses. And then we have our seedless vascular plants, which are going to be our lycophytes and ferns. And then we get into our gymnosperms and angiosperms. One thing to note is that a main part of this lab is looking at vascular tissue in plants. And so it's good to note that vascular tissue originated here. And so our non-vascular plants will not have the tissue types that we're talking about today. One other thing to note is today's lab, we are specifically only talking about our angiosperms, and we're going to be looking at differences between two different groups in angiosperms. The two different groups that we're going to be looking at are our dicots and our monocots. And so here we have our early dicots, and then we have a split between the monocots and the eudicots. You can basically forget about the early dicots for now. We're going to be looking at monocots and our eudicots. And all of these groups encompass our angiosperm group that we looked at. So there's just groups within groups in plants because there's so many. A couple things about dicots and monocots before we move on to different cell types and stuff is that monocots, which is represented by corn in this diagram, and dicots are represented as beans in this diagram. These are the plants that you'll be looking at in lab. So it is nice that I found this this diagram. There are a couple main differences that you'll see in dicots and monocots that hold pretty true to those two groups. The first one being the number of cotyledons that come out of the actual seed. And so when I say cotyledon, these are our seed leaves is sometimes what they're called. And so they are the first leaves that will come out of the seed. And so in monocots, mono meaning one, there's only one leaf that will pop out of the seed. And so that's kind of depicted here in our corn. And then with our dicots, di meaning two, we have two cotyledons in dicots. And so those are represented here. Another main difference that you'll see is that occurs in the venation in the leaves. And so in our monocots, we have parallel veins. So these are plants like lilies. Lilies have parallel veins. And then in dicots, there are net-like vein systems. And then one other difference that we'll just mention today, because you guys will see it, is the different root types that these groups of plants have. So in monocots, we have this fibrous root system, which I believe in your packet, it's called adventitious roots. And so these are roots, these are root systems that are that are only found in monocots, sorry. And then in dicots, we have a taproot system. And so we'll have a main taproot that goes down with a bunch of smaller roots that come off of it. We'll look at the differences between these two in lab, but it's just good to note that monocots do not have a taproot system. Okay, so after going into dicots and monocots, the main thing that you're going to be looking at in lab is the different ways that tissue systems are organized in monocots and dicots. And so just a brief reminder about these, or just a brief what these systems are, is we have three systems that matter, and I'll minimize myself here so you can see this. And so these three systems are present in every part of the plant. In lab today, you'll just be looking at the stems, so which we see here, and the roots. We're not going to be looking at the leaves. But there are three types of main tissue systems. So we have dermal, which is the single layer surrounding the plant, which is this blue layer here. So the main function of this tissue system is to provide protection. The ground tissue system, which we see in yellow here, makes up the bulk of the plant and is responsible for photosynthesis and storage. It's good to know. And then on this slide, I also have where or how these systems arise. So ground meristem gives rise to our ground tissue. And then we have our vascular tissue, which provides support and transports water, nutrients, and photosynthetic product between the root system and the shoot system. And this arises from the procambium. And so our vascular tissue is seen here in purple, and you can see it differs between the root and the stem. And so we'll be looking at that in lab today, along with the differences that come with being a monocot and being a dicot. A couple other things you might want to note is the different cells that make up different tissues. And so we have a bunch of different cells that I'll talk about briefly. First, our parenchyma, parenchyma, and sclerenchyma are all ground tissue cells, and they all serve a different function. So parenchyma are unspecialized cells that carry out most of the plant's metabolism. Parenchyma also functions in support, and the sclerenchyma mainly consists of dead cells that have primary and secondary cell walls, which also provide support. So these are all again, a part of the ground system. Now, and then, so the other two types of cells that you will be mainly focusing in on in lab, I can put my little video back, in lab are the xylem and the phloem. And so I believe we've talked about this, but if not, xylem transports water and minerals through vessel elements and tracheids. So transporting water for xylem, and then phloem is what transports our sugar and other photosynthetic items. The xylem and the phloem are both vascular tissues. So that is good to note. If you want to know more about these, I would definitely also take a look at chapter 37, again, the plant form and function. They have like a paragraph on each one of these cell types. Again, you will only be asked to distinguish the xylem and the phloem. However, it is fun to know what the other cells are too. Here we have, and in the next photo, we'll have kind of a representation of what you're going to be looking at. And so here we have our xylem and our vessel elements, which are, again, carrying our water. And then we have our phloem, which are the xylem are pretty big. The phloem is much smaller, which also have sieves, or the sieve tube elements will surround the phloem cells. And there are also companion cells wedged in here for support. One thing, I guess, one general thing to know or to note is that xylem are going to be much bigger and phloem are going to be much smaller cells. Here in this next picture, it's kind of more zoomed out to look at the differences in the dermal, ground, and vascular tissue. So again, the vascular tissue are these, the phloem and the xylem. The phloem and the xylem will form vascular bundles in dicots, and in monocots. They're just arranged differently. And then yellow is our ground tissue, and then our dermal tissue, which again is our epidermis is around the edge. It's hard to see that, but those are our three dermal or our three tissue systems. And again, we're going to be looking at those today. Okay, so after all of that, the goals of this lab are to be able to identify, for the first activity, to identify the main anatomical differences between monocots and dicots. So you'll look at your corn and your bean plants, you'll draw them, and you'll label some external structures that we want you to know. These are the external structures that will have you identify. Again, all of this information will be in the lab for you to look at, but it'll be a good idea to look at these external structures and make sure you know what they are, where they are, etc. We'll go through a few of them. So we have our stem, our roots, which should be all pretty self-explanatory. So we have our stem here. You can take any portion of the stem, it goes all the way up. Our root system, leaves, pretty self-explanatory. We have here, we have our apical bud. So the apical bud will be up at the top here. Our axillary buds are going to be here on the side. Nodes, axillary buds are lateral stem growth. I guess I could say that. Nodes are stem tissue from which leaves, branches, and flowers grow. So this would also be a node here. Internodes are the distance between, or stem tissue between the nodes. So there's a node here and there's a node here, and the distance in between that is the internode. And again, just the different types of roots that we'll talk about more in lab. Activity 2 and 3, we're going to be looking at the differences and cell types in the root systems and tissue systems between the dicots and monocots. So we'll have you look at some internal structures, which are listed here. So you'll have to identify the xylem and you'll have to identify the phloem. The dye that we're using in class is this toluidine blue. And so it will dye the lignified tissue, which is our xylem, a green bluish color, and the non-lignified tissue, which is our phloem, a red/pink purple. So that's one nice thing. The vascular bundles, which I might have just said and not defined, said and not defined, is the collection of xylem and phloem. And then we will ask you to identify the epidermis, which is just that outermost layer of cells meant to protect the plant. The cortex, which is the cells between the endo and the exodermis, and our endodermis, which is the layer of cells just inside the cortex. And so again, we'll have things in the lab to help you identify all of these structures, but it is a good idea to familiarize yourself with them. Okay, and then the last activity, after you do all of that, you're going to do a short investigation with different stains. So we have about 4 different stains in the lab, which are listed here. We have 4 different dyes that will dye 4 different things in plants. And so for example, we'll have dyes that dye for starches, proteins, lipids, and lignin. And so your goal during this is to pick a plant and a part that you want to investigate. We have carrots, we have celery, and then we'll have a bunch of other things that are popping up outside that you can look at. And so your job is to do 4 different microtomes of this plant, or thinly sliced cross sections, and you're going to be hypothesizing what structures or cell types the dyes will affect. And so we'll tell you what the dyes dye, or what the dyes will stain, I should say. And then you'll be your goal to try to figure out what is going to stain. And that is basically it. Everyone will have their own microscope to work at. There are stain stations at each of the desks. And your TA will let you know a little bit more about how the lab is set up. But for that, we are done. So I will stop this.